So I'm ready. So hi everyone, and thanks Maripat for the kind introduction, and also thank you to Susanna for organizing this meeting. Thank you all for joining me to this talk, in which I will show you why carbon isotope studies are a powerful tool for understanding past and also present geological and biological processes on Earth. As you know, carbon is an essential element, is the main constituent of the biosphere, and its element is quickly recycled in the ocean atmosphere system. So when we study carbon isotopes, we can trace the movement of this carbon in the environment over time, and this is what we call the carbon cycle. So I hope that by the end of this presentation, you will have a better understanding of the importance of the carbon isotopes and the role that these isotopes have in improving our understanding of the Earth and other planets' processes. So let's start. The first question I would like to answer is, what is the carbon cycle? How did it work on Earth in the past? Secondly, how are the carbon isotope signatures on Earth and how are on Mars? And finally, how carbon isotopes can be used in the samples that the Mars sample return mission will bring back home to the Earth. So I will start with a very basic knowledge about carbon isotopes. This information probably is familiar to you, but I think it's worth it to mention. So isotopes are elements that have the same number of protons, but different number of neutrons, resulting in different masses. Okay, carbon in particular has two stable isotopes, which is the carbon-12, which is also called the light isotope, and carbon-13, that is called the heavy isotope because of the mass. But there is also one radiogenic isotope, that is carbon-14. Most of the Earth's carbon, it's almost 99.9%, happens as the light stable isotope, and the remaining 1.1 occurs as the heavy stable isotope. Variations in these isotopes, the ratio between the light and the heavy, are reported in a standard delta notation in parts per mil. Here is the equation, and is derived by the following equation, in which we measure the ratio of the heavy versus the light in the sample, and we compare the heavy versus the light in the standard, where the standard is an international reference material that in this case for carbon is the Vienna PD belemnite, that is this fossil that I represent here in this picture. This standard has a value of 0. So everything is compared, every carbon isotope is compared to 0, is the standard value. So in general, delta carbon-13 in natural samples on Earth range from about -100% to +50%. But however, most of them are in the range between -35 to 5 per mil. But when we talk about carbon isotopes, we are not just talking about numbers. We are talking about processes and geological, biological and geological processes that produce a specific signature. So for example, positive values, which means higher than 0 per mil, we can find different processes, like for example, increasing biomass, decreasing oxidation. But among all these processes, the most the process that exerts the strongest impact is the increase in inorganic carbon burial. I don't want to go too much in detail because it's quite complex, but what I wanted to know is that every time, or most of the times, that we have a positive value compared to the standard, means that there was in the environment an increase in organic carbon burial. And this, the reason why it's positive is because photosynthesis is an enzyme-mediated process that preferentially takes the light isotope instead of the heavy, because it's much easier to break the bond in the light isotope. So it takes less energy to break this bond. So, then photosynthesis and the creation of organic matter takes the carbon-12 and then leaves behind the residual carbon enriched in carbon-13. So if we apply these values to the ratio, then we always get positive values. But also we can get the negative values of the delta carbon-13, and these processes that lead to these negative values are usually the opposite to the positive. So, for example, we can get negative values if there is a decrease in biomass production, if there is release of CO2 from the mantle, or methane that can be released from gas rich in the ocean. But the same, the primary driver is the oxidation of organic carbon. So in this case, what happens is that the organic matter that it's rich in the light isotope is released to the environment. So then there is an increase in 12 versus 13. So the ratio in this case is inverse, and then we get the negative values. But this is mostly the important information that I want you to get from this. Most of the time, positive values are related to increase in organic carbon burial, and negative are related to oxidation of organic carbon burial. And importantly, these negative and positive values are commonly observed in the record, in the rocks. They are very easy to measure. And these shifts between them are considered to reflect this change in the balance between carbon burial and oxidation. So now that we know what the carbon isotopes are, we will use them basically to trace the carbon cycle on Earth. So if I show you a picture like this, you will probably think about idyllic and relaxing holidays, the ocean, snorkeling, and could be. But the reality is that in this picture and on Earth, what is taking place is that carbon moves in a complex web of interactions through the different carbon pools. That on Earth are the atmosphere, the hydrosphere, the geosphere, and the biosphere. These interactions are dynamic, and changes in one pool can affect the size and functioning of the other pools. So in the carbon cycle, there are many processes involved, but I would like just to mention the most relevant, which are the carbon fixation so carbon dioxide from the atmosphere and water together are producing organic matter and release oxygen. Respiration, that is just the opposite. So organisms release carbon dioxide back into the atmosphere by breaking down organic matter to produce energy. Organic matter decomposition, that is breakdown of organic matter by microbes. And carbon sequestration, that is the process of storing carbon in long-lived reservoirs, like, for example, rocks. So the way that we have to trace the carbon movement in this among all these processes is by using this carbon isotope. But importantly, what I would like to highlight is that most of the processes that I just mentioned are related to biology. And that's why isotopes are one of the most extensively used biochemical tracers of biological processes. The carbon cycle works at equilibrium. It means that it's a closed system that is maintained at equilibrium in the long term. In the long term, I mean the geological long term. Because in closed systems, we can apply the isotope mass balance concept to track the evolution of carbon fluxes between these reservoirs. And this concept, the mass balance concept, basically says that in the long term, it is assumed that there must be an isotopic balance between the isotopic composition of carbon that is entering and exiting the atmosphere-ocean system. So everything that enters, every carbon that enters in the system must be equal to the carbon that exits the system. So these diagrams, briefly summarize, is much more complex than this, but these diagrams summarize very briefly the carbon source and sinks. And basically, the carbon enters in the system from the mantle in the form of CO2. Part of the CO2 will form carbonates here, and the other part will be used by photosynthesis to produce organic matter that will be finally buried in the sediment. So the total carbon will go to the atmosphere again, through weathering or metamorphism, or it can go to the mantle through the subduction, getting finally a closed system. But what is very interesting from this explanation is that the relationship between the source and the sinks on the geological time scale is determined by the amount of carbon that is converted into organic carbon, that is the fraction. That's that fraction here. So this is the carbon that enters in the system. This is the mantle. This is the organic carbon that is stored in sediments, and this is the isotope signature of the carbon that is stored in carbonate. So the isotopic composition of the Earth's mantle is generally assumed to have been constant through time, that is -5 per mil. But we also know the carbon isotope compositions of organic matter and carbonates, that is on average -25 per mil and 0 per mil respectively. So using these values in the equation, so we put here the mantle, the organic carbon in sediments, the organic carbon in carbon in carbonates, we can derive this fraction here, and we get a number of 0.2. And 0.2 in this fraction means that 20% of carbon today is buried as organic matter, and the remaining 80% is buried as carbon. So this system, this closed system, is known as the long-term biological, biogeochemical carbon cycle, where small changes in the volcanic or metamorphic carbon input, in any of the inputs in general, and the combination of weathering and organic carbon burial usually modulate atmospheric CO2 levels and then regulates the climate over time. But I have a question. But what happens when sources are not equal to sinks? What happens when the system is not balanced, when this is not a closed system? So what we have is that we find perturbations in the carbon cycle. These perturbations, these variations in the carbon cycle, are also known as excursions. So if I refer to them to perturbations or excursions, I'm talking about the same thing. So this graph shows the delta carbon 13 from the Archean to the present. Is separated by in colors depending on the eras. And then I have added in the background the different phases of atmospheric oceanic oxygenation that took place in two main steps. That is the great oxygenation event and the neoproterozoic oxygenation event. During the GOE, oxygen increased from 10 to the -5 to the 10 to the -1. And during the NOE, the NOE, it increased from the 10 to the -1 to modern values. It's comparing in the present atmospheric levels. This is the unit. So when we have this picture here, we can highlight different things. The first one is that we can observe that the delta carbon 13 values of marine carbonates have remained relatively stable between -5 and 5 per mil. And the second thing is that the main perturbations, the most positive here and the most negative here, are occurring at the end of these periods of great oxygenation event and neoproterozoic oxygenation event. And the third point that I would like to highlight is that after those dramatic increases in oxygen on Earth, these dramatic perturbations of the carbon cycle, two of the most important biological innovations took place, that were the emergence and diversifications of eukaryotes and animals, respectively. So let's see a little bit more about these two big perturbations, these two excursions, because I think it's worth it to show you what I'm talking about. So the most positive one, with values that reached up to 10 per mil, is known as the LOMAGUNDI excursion. And it occurred around 2.2 billion years ago. And as we saw in the previous slides, positive values are related to increase in organic carbon burial. So actually, this excursion is thought to be driven by an increase in inorganic carbon burial. This excursion was first identified in Zimbabwe, but subsequent studies revealed that it was a global phenomenon occurring in sedimentary rocks across the world. And this fact is important because we can have an idea about how the Earth system, with the interaction of these 4 carbon pools, work in the long term. It's a global phenomenon that covers the whole Earth. However, the reason of this excursion, the LOMAGUNDI, the drivers are still a topic of debate because there are 2 principal hypotheses. There are several, but 2 of them are the most relevant ones. The first one suggests that it was driven by an increase in photosynthetic productivity. As I said, increase in photosynthetic productivity means that there are more organic material in the ocean, and ultimately this organic material will be buried and preserved in the sediments. The second one says that it could be triggered by tectonic activity. That it could be, because if you see here in the right hand graph, all these segments represent different supercontinents assembly. So this continent here was Kernorland and was formed at that time. So it's real that there was a strong tectonic activity, and this activity led to change in the ocean circulation and nutrient availability. However, why not a combination of both hypotheses? Why? Because note that here the oxygenic photosynthesis started quite before the oxygenation event. So at some point, around 2.4 million years from here, the atmosphere started to accumulate oxygen because the production of this oxygen exceeded the consumption by organisms. So this oxygen production was likely driven by tectonic activity that led to erosion. When we have erosion from the continents, it means that a lot of nutrients will finally go to the ocean and to the water bodies in general. These nutrients will be likely fuel increased primary productivity. So we will have more organic matter in the system. So more organic matter will be finally buried in sediments. My opinion is a combination of both could be the drivers of why this excursion was so negative, and also took so lasted for so many years in the system. But now let's go to the second one, the most negative. So we have seen the most positive, and now let's move to the most negative. And this one sounds very familiar to me because I did my thesis on this topic, on the SHURAM excursion. This excursion, the SHURAM, is the most negative in Earth history. It was first also described in SHURAM in Oman. And then, as in the LOMAGUNDI excursion, correlative studies from around the world showed that this excursion was a global phenomenon. So to get a better idea about the SHURAM, it's important first to understand the geological and biological context that led to this excursion. So during this time, the Neoproterozoic era, 3 different global glaciations took place. These global glaciations means that the Earth was completely covered by ice. And they are usually known as the snowball Earth. And this fact, this process has slowed down the carbon cycle dynamics. Also, there was an important reconfiguration with the breakup of the continent Rodinia, that was some million years ago later, was assembly in the Gondwana. So remember that when we have tectonics, we have nutrients in the system, we have erosion, we have nutrients in the system. Remember that. And lastly, there was a period during the Neoproterozoic that shifted from oceans that were predominantly dominated by bacteria to oceans dominated by algae. So under this scenario, the carbon cycle witnessed a very strong and global reorganization, reconfiguration. And in South China, that this picture is in South China, it's an area that I studied during my thesis. I collected samples from these outcrops. And so in this, in South China, this is the signature of the carbonate carbon isotopes and the organic carbon isotopes. And then we can see that here there is a sharp shift from zero or positive values to negative values. So to understand these negative values, there are also different hypotheses. The two most relevant talk about the diagenetic effect. Diagenesis is when you have the rock has been deposited and post-depositional processes may change these rocks, the isotopic signature or the stratigraphic or sedimentary structures that were originally formed. But also the second hypothesis says that at that time there was a massive amount of organic carbon in the system that was episodically oxidized. And remember that oxidation of organic matter usually leads to negative carbon isotope excursions. And this situation, this oxidation, was maintained in the system until it was balanced here because new primary productivity drove the excursion to near zero or even positive values. Then this primary productivity made the system to return to at equilibrium. So as I said, I worked quite a lot on the SHURAM excursion, and personally I support the second hypothesis. I have many reasons to support them, geochemical of course. My work in this SHURAM excursion consisted on exploring the drivers that promoted the recovery from this SHURAM to equilibrium. And then is when I concluded, based on nitrogen and Raman spectroscopy, several proxies that increased primary productivity led the system to equilibrium. And this primary productivity and the burial of organic carbon usually releases oxygen to the atmosphere. And then this fact is important because the SHURAM excursion and this oxygenation might be linked to the onset and diversification of animal life. The SHURAM, here the SHURAM excursion, brackets a major transition in biological innovations because it happens immediately after the global extinctions of large architects, which are these microorganisms here, but prior to the Cambrian explosion. So here during this period, the Ediacaran biota emerged, and then there was a diversification, but suddenly they disappeared just before the Cambrian explosion. But there is an increase in the complexity of life. So that now that we have a glimpse on the carbon cycle, which I find really fascinating, how we are able to extract so many information just from one data, from one piece of sample, 1 gram of sample. How can we reconstruct the whole history of an area on Earth? So now that we have this idea, let's see the broad picture of carbon isotopes on Earth and on Mars, and also how the carbon cycle operates on Mars, if any. So this graph on the left summarizes the ranges of carbon, delta carbon 13, of geological and biological reservoirs on Earth. Geological are here represented in black, biological are represented in green, and I intentionally, I've left another geological in yellow that I will explain a little bit later. So from this graph, we can extract some conclusions. The first one is that the geological delta carbon 13 range is usually between -10 and +10, which is consistent with the biotic process, as previously seen in the excursions that I've mentioned before, the LOMAGUNDI and the SHURAM excursion. Also, the biological range varies between -2 and -85 per mil, which is consistent with biotic processes, because these biotic processes, as I said when I mentioned earlier the photosynthesis, processes preferentially take the light isotope and then leave the environment, the residue, the residual carbon enriched in carbon 13. So we usually get these low carbon 13 values. But what is remarkable here in this graph is that, for example, methane, that we can see down in this graph, can produce very, very negative values. Like, for example, these methanogenic bacteria. Methanogenic bacteria are the microorganisms that can generate methane from organic sources with values down to -40 per mil. But we can also see methanotrophic bacteria that are the organisms that utilize methane as a source of carbon. And in this case, they can reach values as low as -85. And also, I think it's important to highlight these examples because methane on Earth via methanogenesis is almost entirely, directly or indirectly, biologically produced. And the presence of isotopically light methane is commonly assumed to provide a signature of microbial processes. However, abiogenic serpentinization, that is the yellow color that I left here at the end, can also produce methane with high negative delta carbon 13 values, as low as -54. So this abiogenic serpentinization is a chemical process that happens when we have certain types of rocks, like igneous rocks, and these rocks are exposed to fluids, and these fluids are rich in hydrogen and methane. But biology is not required for this process. And there is another example about methane, that is the oxidation of methane, that is generally driven by aerobic oxidation. But however, this oxidation, methane, can also be linked to oxidants, like, for example, ferric oxide or nitrite or even sulfate reduction, that can be formed in an anoxic ocean atmosphere system. So, consequently, carbon isotopes are a very, very powerful geochemical tracer, but there is often more than one explanation for the delta carbon 13. And I think that this is a very important message from this seminar. So whether the end goal is to trace the carbon cycle or find evidence of biological activity, we need a strong geological context to derive a unique explanation of the signature that we are getting. When we work with carbon isotopes, geologists or geochemists, we work always with additional data, with supporting data that may provide robust evidence about what we are looking at. So we need to build a comprehensive geological model for the area that we are studying, and to get this geological model, we integrate data from different sources. For example, additional isotope data, iron, oxygen, nitrogen, sulfur, a lot of isotopic data that all of them will give you a clue, and all together will give you the whole story. But also bulk geochemical data like phosphorus or redox sensitive elements. and also organic matter mineral associations, because when we look at the rocks itself, we can see what's the mineralogy, what's the petrology, the kind of sedimentary structures, if there is any alteration post-depositional, and all this information gathered together help us to understand the story of why we are getting these carbon isotopes signatures. So now if we move to Mars, because all this carbon data was on Earth, but if we move to Mars, is there a carbon cycle on Mars? Well, if we think in a carbon cycle on Mars that is operating in the same way as on Earth, the answer is no, at least in the present. In the past, the past carbon cycle on Mars is still a subject of scientific investigation, because we don't know the things and the sources of in the past of this carbon. So the principal difference is that Mars has no biosphere or hydrosphere, which are critical components of the carbon cycle as it works on Earth. So the transfer of carbon is limited to the atmosphere and the geosphere, atmosphere and geosphere. And there are no significant mechanisms that regulate this carbon dioxide in the atmosphere. So this situation has contributed to the current state of Mars. That is a cold, and as you know, it's a cold and inhospitable planet with a thin atmosphere that is mostly composed of carbon dioxide. So like the carbon cycle on Earth, there are sources and sinks of carbon on Mars. The source is exclusively from the magmatic outgassing. Then there is a free carbon reservoir formed by the regolith, the polar caps, and the atmosphere that they interact among them. And the principal sinks are the carbonate formation and the atmospheric escape through primarily photochemical escape, and secondly, pickup ion sputtering. The principal sink is the escape, because carbonates are not very common on Mars. Carbonates have not been detected, have been detected, but just in a few spots on Mars. So the principal sink is through escape. So under this different concept of the carbon cycle on Mars, it's not surprising that the carbon isotope values reported from the literature are so different from the ones that are measured in Mars. And here in the next slide we have the comparison. When I was preparing this presentation, I came across this picture on the left on the delta carbon 13 of different objects in the solar system. And I found it very illustrative, because without having a deep knowledge about carbon isotopes, it's pretty easy to observe that the carbon isotope signatures of the different objects are impressively homogeneous, and they overlap with the average carbon isotopic value of Earth's mantle, that, as I said before, is -5 mil. So it's around this range. And this happened for all most of the objects in the solar system, except the atmosphere on Mars. But you can see that is quite shifted compared to the rest of the objects. So if we look at the graph on the right, we can see that the isotopic ratios are also very different. I think I can overlap. Yes. Those are the orange shadow is the terrestrial range, overlapping the Martian range. So looking at this graph, we can see that the isotopic ratios are significantly different compared to Earth, and it could indicate two things. Mars, the carbon cycle on Mars is operating in a very different way, which is not surprising. Because there is no atmosphere biosphere or hydrosphere on Mars. But also, it could mean that there are other factors that play in that are affecting the isotopic composition of carbon planet, but we don't know yet what these factors are. So I have organized the Martian delta carbon 13 values. I divided into meteorites, atmosphere, and in situ. The first observation is that the mantle value here is far below the Earth's value, that is -5, and here on Mars is -20 per mil, yes. However, this value was derived from meteorites, so we don't know if a real measurement from the mantle would give the similar signature. Then delta carbon 13 from the atmosphere shows a wide range. Here in the Viking, measured from almost 30 per mil to almost nearly 80 per mil, which is significantly higher than the atmosphere on Earth, but it's also not surprising as the chemistry works exactly the same as on Earth. So the light isotope is escaping easier than the heavy isotope in the Martian atmosphere. So it makes that this atmosphere is enriching in carbon 13. But the last set of data comes from relatively modern analysis, all of them carried out in the Gale crater, and also with delta carbon 13 values higher than terrestrial samples, even when we compare different lithologies, like for example here, comparing sandstone, mudstones, and aeolian deposits. But the most striking observation is this one, is the low carbon 13 content in methane that was measured with values down to -140 per mil. That this range overlaps biological processes on Earth, as we can see in this orange shadow. This data was published, I think, is in 2022 or 2021, I don't remember, but very, very recently. And this publication brought about high expectations on how could be the potential origin of this sample. But as I mentioned before, there is often more than one explanation for the delta carbon 13 values. And in this case, up to 3 different theories could explain the presence of this low carbon content. In this study, authors referred to the cosmic dust as one of the hypotheses. A second one was the ultraviolet conversion of CO2 into simple organic compounds. And the third one, that is the most exciting, is the hypothesis that says that microbes from the Martian past consume methane that was released into the atmosphere. And they got this conclusion compared to light and heavy isotopes on Earth. But up to date, we don't have a record of past surface microbes. So we need, it's necessary more evidence, more analysis, more studies combining different techniques to finally agree or disagree with this conclusion. So for the moment, there is no life on Earth based on this carbon isotopes analysis. So in any case, these signatures that we saw in the previous slide and the next to come necessitate further investigations and additional geological and geochemical analysis, because we need to elucidate what are the carbon sources that are originating these signatures. And this is at this moment is when the Mars sample return mission comes into action. This mission developed a series of scientific and engineering objectives from the scientific point of view. The objectives are divided into 7 that include the geological processes, biological history, evolutionary timeline, volatiles, how can be the interior of Mars, the environmental hazards, and also resources for future missions. But among all these objectives, the biological history comes with 3 objectives. That in this case is to study the carbon chemistry on Mars, ancient biosignatures, and modern biosignatures. So, these sub-objectives, the carbon chemistry and the reality is the carbon isotopes play a crucial role in improving our understanding of the carbon cycle on Mars and the potential habitability. So, carbon isotopes are a main objective of this mission, of the analysis, the samples that this mission will bring back home. Just an update about the cores that are being drilled on Mars in the G0 crater. The objective is that the Mars sample return will bring back home up to 38 samples. Up to date, 19 cores have been taken successfully and stored. One is from the atmosphere, so actually there are 18 cores. One is from the atmosphere, 2 from the regoliths, and 16 rock cores. The collected rocks are igneous and sedimentary, but they belong to a variety of environments spanning igneous floor, delta front, upper fan, and marginal carbonates. And this variety of environments and lithologies make this mission highly attractive because from the geological point of view, the variety of samples will allow a comprehensive geological and geochemical characterization of this area that will help us to understand and contextualize results when they come. So nowadays, Mars is cold. I mean, it's an inhospitable planet with a thin atmosphere. But in the past, Mars is believed to have had a more friendly environment billions of years ago with liquid water on its surface and a thicker atmosphere. So these conditions could have potentially supported the existence of ancient microbial life that may have interact with carbon in a similar way that is occurring nowadays on Earth, in the carbon cycle on Earth. So these samples from the Mars sample return have strong implications. Strong implications in our knowledge of the potential habitability of Earth on Mars. Sorry. So these carbon isotopes are expected to be a valuable tool for determining the habitability of Mars. But some aspects in which carbon isotope studies may help determine this, trying to determine this habitability of Mars are, for example, determining the presence of organic carbon. So if the carbon isotopes composition of the organic carbon is consistent with biological activity and is supported with additional studies that also confirm that the signature has to be biological, then it would suggest that the life on Mars could have existed. Also, another implication is the study of the Martian carbon cycle, because carbon isotopes can help to better understand what are the sources and sinks of carbon on the planet, and also how this carbon is moving through these sinks and sources. Also, the carbon isotope studies may help in identifying the source of carbon, because it's very important to know if this carbon is derived from biotic or abiotic sources. And this is, as I said earlier, that carbon isotopes, together with additional isotopic studies or bulk studies, may provide us the answer. And also another implication is the understanding of the potential for contamination. And this topic is very challenging and is very important. Because carbon isotopes can help to identify any potential contamination of the Martian samples with terrestrial material. So we can discriminate if the samples, when they arrive to Earth, we can discriminate if the signature is proper from Martian samples, so it's contaminated. And we need this information to ensure that the results of the carbon isotopes are accurate and reliable And the objective is to avoid the misinterpretation, is to be clear that the signature that we are looking at makes sense and is not overlapped by additional material, in this case from Earth. However, there are also limitations that may directly impact on the carbon isotopes studies. And these limitations we have to consider and anticipate, if possible, because these isotopes, carbon isotopes, will provide valuable insights into the sources and processes involved in the formation of carbon. But some limitations are, for example, the limited sample availability, but it's normal. I mean, the rover has characteristics and has some physical and limitations, and also the mission, the Mars sample return mission, has technical limitations, and we will get up to 38 cores, drill cores, but this amount of material is limited, and so we have to be careful, because when we study rocks, if we have just just a little material of the sample that we are studying, it can make difficult to draw robust conclusions about the results. Another limitation is the contamination, but in this case is because the Mars, the Martian surface, is exposed to cosmic radiation and also to solar wind, which can have an effect in the carbon compounds present on the surface. And also, as I said, the contamination of Martian samples with terrestrial carbon that can lead us to erroneous conclusions. Also, another limitation is the uncertainty that we have nowadays about the carbon cycle, because it's not well understood. There is a considerable uncertainty about the sources and processes involved in the formation of carbon compounds, and this uncertainty make it challenging to interpret carbon isotope data, specifically if multiple sources and processes are involved. Ideally, we should be able to characterize all the sources independently. But if we have a sample that, in a way that if we get a sample with different sources, we can discriminate between the different origins of this carbon. Also, another limitation is that there is a small isotopic variation. The carbon isotope variation, the ratio between the heavy and the light that is observed in Martian samples is relatively small compared to terrestrial samples. And this can make it difficult to differentiate between different carbon sources or processes based on this data. And finally, another limitation is the complex geology that we have in Mars. So the formation and alteration of carbon compounds can be, in this case, affected by various geological processes such as weathering, volcanic activity, or most commonly impact events. So these processes can result in the mixing of carbon compounds from different sources. And as I said before, if we don't have a proper characterization of the individual processes, the signature that is produced in individual processes, is much more difficult to identify these different sources in one sample that contains different carbons. So at this point, I'm concluding this presentation with some takeaway messages. I tried to summarize in four the four most important takeaway messages. The first one is that carbon isotopes are critical to understand the coevolution of life and environments over the Earth's history. It's as I said before, in just a few grams of sample, we can reconstruct how was all the paleo environment, and how were the chemical conditions in the column water, for example, that facilitated the production and preservation of organic matter. So these isotopes can give you a whole story that, supported with more analysis, can give us the full story. The second takeaway message is that this delta carbon-13, the carbon isotopes, are traces of biological processes, but interpretations must be supported by additional analysis. Also, the modern carbon cycle on Mars is still poorly understood. This mission, the Mars Sample Return mission, will likely will likely provide some answers to the different questions that I personally have nowadays. That is, how was there a past carbon cycle on Mars, and if so, how was this carbon cycle? Was also the interaction of 4 different pools of carbon like on Earth, or it worked differently? And even if it worked differently, it was possible life. So life adapted to how carbon moved in a different way on Mars. I have a lot of questions about what happened on Mars in the past, and I think that carbon isotopes are key to understand not only the present but also the past on Mars and also on other terrestrial planets. So from my side is everything. So thank you very much for listening, for staying connected to this talk. I know it was a lot of information in 30-40 minutes, but I think that you enjoy and you get a quick, brief glimpse of what carbon isotopes can tell us about the Earth, about Mars, and about the evolution of both planets. Thank you very much.